welcome everybody to today's presentation on neuron and core neuron. I am Robert McDougall, and with me today,
myself, Pramod Kumar.
All right, so basically, I was just going to do a brief intro to neuron. It's not a tutorial, but it's an overview with a bunch of examples. And then Pramod is going to take us on a tour of some of the newer features and where we're going. Does that sound fair, Pramod? Do you have anything? All right. That then being the case, let's get going. So we're going to start with just a brief tour of neuron. So basically, as where we're going here, we'll just, what is neuron? How do we use and how do we learn to use neuron? What are the core concepts that are involved? Well, then we'll look at some examples, talk about working with data a little bit, and highlight a few third-party tools and resources before we move on. All right, so what is neuron? I like to think of neuron as a multi-scale framework for representing neural simulations. And those neural simulations can be on any of a variety of scales, and that's sort of one of the things that makes neuron great. We have interactions that could be done at the level of integrate and fire cells, where all you really have is spike timings. Neurons' claim to fame is a lot dealing with morphologically detailed cells and networks of morphologically detailed cells. But certainly we can zoom in and look at three-dimensional chemical signaling within the cells. We can zoom out and look at extracellular interactions. So here we have a spreading wave of elevated extracellular potassium that's propagating throughout a certain volume of space. And so we can do all of these kinds of simulations. And the nice thing is you can sort of plug and play and mix all of these different levels of biophysical complexity. But that's really one of the things that makes it possible for us to do this is that neuron does, in fact, focus on trying to link everything to biophysics. So there's an action potential. There's, at the very least, even with integrating fire, there's a spiking event. Units are important, right? We talk, these are not generalized abstract mathematical models of the brain. We focus on normalized position, distances in microns and things like that. So all of the sort of biophysical units that we can make these levels of complexity and sort of plug and play, mix and match, and see how all the pieces go together. We're not going to talk about it today, but I do want to give a highlight. So neuron does have a graphical interface that is available if you're running it locally. It's not available if you're running it on eBrains. So we're not going to go into it today, but it does have a graphical interface that allows you to do a lot of model introspection, a lot of model analysis. And even you can build some models, load stuff in, do exploration, decide, does this 3D morphology represent what I think it represents? So there's a lot of power that you can get from just using the graphical interface. If you're interested in that, we have plenty of tutorial videos that go hours and hours of tutorial videos that go into using the graphical interface. I'll give you the link in just a second. But in addition to this, so neuron's been around for a while. The ideas behind neuron go back to a 1984 paper by Michael Hines, who has always been the lead developer for neuron. And over the last almost 4 decades now, neuron's found use in over 2,600 publications that we're aware of. We don't have the source code for all of them, but if you go on ModelDB, you'll find, I should update this slide, as of yesterday, 800 published neuron models that are available on ModelDB. All of this stuff is fully Python scriptable. There are other languages that neuron supports. You can interact with it through a language called Hogue. If you're really ambitious, you can try to log. It's a library. You can try to use that library from C++. In the past, we supported Java. There's plenty of things out there that you can interact with neuron, but the most natural for most people is going to be to go in through the Python interface. Like I said, covers a wide variety of levels of complexity. And one of the other great things about neuron is So this runs on a wide variety of hardware. So you can run this on an old single core computer if you can still find one of those. Or we support threading, multiprocessing, both locally on your individual machine and on HPCs. Thanks to the bluebrain project over the years, we've had the opportunity to play around with some really large toys and make sure that Neuron scales well on that. We'll talk about GPU support with Core Neuron in the second half. And of course, you can run this on Google Colabs or eBrain. So if you want to just pop up your cell phone and do some neural simulation, that's always a thing. All right. So how do we use it? How do we learn to use it? By the way, if anybody does have questions, please feel free to post them in chat. But so how do we use it? How do we learn to use Neuron? The central resource for all things Neuron is our Read the Docs page, which is at nrn.readthedocs.io. So you can go there and you can find information on installation, downloads, and instructions. If you're using most of the cloud providers or Linux or Mac, it's a one-line install to get things up and running. If it's Windows, you have to download an installer, but it's not much harder. Just be agreeable as you go through the installer and you'll have everything set up. We do on the left here have a large number of training videos that are available for Neuron. The largest resource of this is from last summer's webinar series. We met twice a week for, I think, 2 hours a week for something like 10 weeks. So there's like 30 some odd hours of tutorial videos that came out of that. They have worked exercises, they have discussions. It starts out with a discussion of the GUI and Neuron features and goes into some depth on a large number of items. And we encourage you to check all of that out. There's also, of course, the guides, which provide a bunch of text tutorials. Programmer's reference, so we have all the documentation is available in Python as well as another programming language called Oak. Links to the Neuron forum for all of your question answers. If you find anything that's somehow not covered in one of the tutorials, plenty of information about who's used Neuron, how you define Python, examples, lots and lots of stuff, all available on Read the Docs, all searchable. If you have any questions, should be your first check for finding things out. All right. So how does one get Neuron going? Well, this is an eBrains meeting. Nice thing is, on eBrains, it's there. Yay! Congratulations. Good job. So in eBrains, the 2207, so you have a variety of kernel pack options when you go to launch a collab on eBrains. If you're using eBrains 2207 or in the pre-release, you're already going to have Neuron 8.0 available. This is fairly recent. It is not completely recent. So I think for the purposes of the core Neuron demos, we'll upgrade to a newer version just to make everything more powerful. The current release is 8.2.1. Even on eBrains, though, this is no big deal. On a single line in a collab, pip install Neuron -- upgrade, and you can give yourself the newest version of Neuron if there is some new feature that you're dying to try. If you're using Google Colab or any other public Jupyter instances that don't have Neuron available, again, same thing, pip install Neuron and you'll have Neuron ready to go. Same thing works if you're using Linux, the Windows subsystem for Linux or Mac OS. And on Windows, again, unfortunately, you can't do pip install for Windows, but you are absolutely welcome to just go to Read the Docs and grab an installer if you want to run things locally. All right. One of the great resources available for learning how to use Neuron is to look at other things that have been done with Neuron. And one of the great resources to find things that have been done with Neuron is ModelDB. If you're not familiar with it, ModelDB is a discovery tool, now has about 1,800 computational neuroscience models enclosed in it. Every single one has an associated publication, so you can go back and explore Try to understand, what has been done with neuron, what context have things been done with neuron. We have associated metadata, so you can find examples of, hey, look, I want to use a function, it's called f-initialize, but I don't quite understand the documentation. Well, then you can go and just search for it on ModelDB and see how other people have used that particular function or class or whatever. And you can do this for non-neuron things as well. So with 800 neuron models, we have lots and lots of examples. You can reuse some of the source code, check out the license terms, obviously, of course, but generally speaking, a large fraction of the stuff in ModelDB is shared with a attribution expectation. But other than that, it isn't. There's terms of use on the site that say basically you get something from ModelDB, cite the paper, cite us. But so you'll go and you'll find, well, we're going to just use, for the purposes of today, we're just going to use mechanisms, ion channels, and things like that are built into Neuron, but these can be specified using a language known as InModal. It's the same language that's used by a number of other tools, including Arbor, which I believe has a simultaneous presentation right now. But so you can go and you can find examples of that. ModelDB will tell you if you find a L-type calcium model, you can click on this. There might be a little star next to the file name. That star will tell you if it's been reused in other models, and that'll give you a sense of, oh, this L-type calcium model has been used in maybe half a dozen things. They were CA1 pyramidal cells and CA3 pyramidal cells. Okay, there's a lot of history of using this particular mechanism with this given cell type. If you're looking at other things, and if you're looking at a Purkinje cell and you don't see that, maybe you want to try a different version of an L-type calcium channel if you think that's important. Right, so this is the other advantage of having these kinds of resources available. You can see how things have been used in the community so far. And I find it to be a great resource for learning about how to use Neuron and other simulator platforms. If you like to read, I do want to give a shout out. We do have 2 Neuron methods papers that came out this year. So one that's focusing on the chemical intracellular signaling, which we're not going to go we're going to have one example that barely touches on that today. But there's papers on that you can check out. And there's another large paper that's sort of a discussion of infrastructure, neuron development, and performance, and where we're going. And you can check out both of those resources if you're so interested. All right, so that said, let's talk about some of the core concepts that we need for Neuron. Well, not really a mental model of how to use it, but there's the core concept. You have to make it go. Oh, that is an unfortunate name. I should not say core neuron for functionality. Sorry, Pramod. But in any case, if you want to load the central neuron functionality in Python, pretty much everything is available through the H submodule. So you would say from neuron import H. And so when we go and we tell it to run for a while, it'll be H dot something. When we go and we construct a current clamp, it'll be H dot something. When we define regions of the geometry or a artificial spiking cell like an integrated fire cell, it'll be H dot something. There are some other things that do come up here. So strongly encourage you to use units in your code anytime that you're trying to model anything and represent anything. So of course, so Neuron, the units are available from neuron.unit, so you can import millivolts, milliseconds, microns, or μm, whichever you prefer, for microns. We support for 2 reasons instead of just having the type μm. 1, because U is easier to type on an English keyboard than mu, and 2, originally Python 2 did not allow non-ASCII variable names, and so this is a historical tradition, a holdover, that we continue to support U instead of mu, but I think it's again a lot easier to type. And also, of course, we support chemical dynamics. If you're doing other chemical dynamics from Neuron import RxD, there's various other things that you might want, but these are sort of the cores, core subcomponents. If you are trying to use things with neurons built-in graphical interface, for example, from neuron import GUI, and there's more. Okay, Neuron itself comes with a large variety of libraries that exist to make your life easier. So there are libraries, and we'll see one for importing 3D morphologies, right? So that's great if you're trying to grab something from neuromorpho.org, which has a vast number of neuron reconstructions with varying shapes. Neuron has libraries for electrotonic analysis, model introspection, load balancing if you're trying to build a parallel model and you want to make sure that you're distributing compute weights evenly across this thing. Neuron has built-in libraries for that. The one that you almost always want, and I think I use it in all of my examples for today, is the STD run library, which just basically provides some high-level functionality that allows us to easily run models for a long period of time. In particular, there's the continue run function. And so h.continue run allows us to run a simulation until a certain time. So maybe I want to measure something at various time points. So I'll initialize my simulation, I'll continue run it until time 10 milliseconds, and then I'll do a sample, and then I'll continue run it until 20 milliseconds, and then I'll take another sample, and then I'll continue run until 30 seconds. And there are other ways, by the way, of streamlining periodic measurements, but that's one approach. All right. So on to more conceptual things, though. So in Neuron, fundamentally, because of in Neuron, a lot of our interest has to do with morphology, right? Neurons have complicated shapes. And when we think of the shape of a neuron, so let's say I've got a pyramidal cell. It looks something like this, right? It has various branches at various points. But it has these large stretches or sections of this, we might say, large sections wherein there's no branch points. And so in Neuron, we call those sections. And so basically you can think of a section as an unbranched chunk of cable and then a bunch of sections. So at a branch point is just 3 sections meeting. And we can connect the sections together, and the sections have various properties. And so this is where all of our dynamics, everything that's happening happens in some section of the cell. Could be the soma, could be some part of the apical dendrite, could be the basal dendrites, could be wherever. So we use section as the keyword for defining these objects that represent parts of the cell. And obviously you can have multiple references to a section. You kind of have to in order to be able to use a for loop. Certain nice things are available. You can print the sections out. More interestingly, of course, sections have various properties, key of which for the concept of the biophysics are there's a length. And so if I have my section that's dendrite, D and D, well, then the length of that dendrite is D and D dot L. The dendrite has a diameter. Well, so this is a little bit tricky because if I define it this way, I say den.diam is 2. Default units here are in microns. Honestly, you're almost always going to want the defaults, but I do encourage you to load in the units and be explicit about it. But in any case, so I can take this and I can define a different diameter. But so these are sort of some key properties of a section. There's also another key property, which is inseg. This is not a biophysical concept, but in order to actually integrate one of these things and to solve something spatially, you have to break it down. You calculate the membrane potential at various points along that section. Because if you think about an axon, right, there may be a very different membrane potential down here as over here, right? It takes time for something to propagate down an axon. So in order to capture that effect, we discretize the axon into pieces. And so if I just have a single piece, Neuron will compute the membrane potential at the center. If I have 3 pieces, neuron will compute it at the center, but also at the centers of the other thirds. One thing that we do suggest, we strongly suggest that you use odd numbers instead of even numbers, because it is often nice to be able to have a node at the center and just to think of it conceptually as being at the center. But so neuron never really defines. You can ask questions about each of these pieces. Each of these spatial compartments is called a segment. A segment is a chunk of a section in neuron, and you can loop over all the segments and you can see where they're computed at. So if I'm looping over all the segments in DEND, when I say that there's 3 segments, well, hey, look, the first one is, well, let's go to the middle one. These are all normalized positions. So the first one is 1/6 of the way through the dendrite, and it has a certain area. The second compute node is halfway through the dendrite, and it has a certain surface area. And the third one is 5/6 of the way through the dendrite, which will be here, and it has a certain area. So we like to represent everything in terms of fractional positions along the segments, along the sections rather, for 2 reasons. One, because it allows us to refine the mesh, and so we can just say, hey, no matter if I'm using 1 segment or 27 segments to capture the dynamics of the axon, I can always refer to a specific normalized position along that axon, and instead of a segment number, right? And so the same spatial location, it should be the same spatial location, no matter how many pieces you break it into. And this is important if you're trying to look at convergence. Have you used enough spatial resolution to actually capture the dynamics of the shape? You can also, the syntax, by the way, allows you to, for the second reason, for using normalized position instead of actual position is that it allows us to explore when things vary. So, hey, look, how would the story be different if it was 10% larger? I'd still want to maybe compare halfway down the section to halfway down the section. But if you don't like that, you could just use the actual position, divide by the length, and then there you go. Then you can think of everything in terms of actual position as well. And from a segment, you can query it. The segments have various properties, including properties about ion channel gating variables and things like that. The one other thing that I really do want to hint at about neuron and our shapes, neuron does not, there's a misconception that some people have that neuron uses cylinders because it's easy to use cylinders in neuron with just specifying a length and a diameter. Neuron actually constructs, allows you to specify arbitrary complexity, combinations of frustums of right circular cones. So in other words, think tapered cylinder and a tapered cylinder and a tapered cylinder and a tapered cylinder. So you can get dendrites of rapidly varying width. So here, this is basically a truncated cone that starts out with a diameter of 1 and goes to a diameter of 5. So this is important for capturing some of the complexity of the shape of the individual cells. If you're doing 3D simulations, Neuron has an algorithm that was published in that 3D methods paper that allows us to convert this into a watertight surface and volume for 3D simulations and sort of join things together nicely. One caveat that I will say, if I didn't specify a length and I didn't specify a diameter, you might have been a little bit surprised because the default diameter in Neuron is 500 microns. The default length in Neuron is, I believe, 100 microns. The default axial resistivity is 35.4 ohm centimeters. And the default temperature is 6.3 Celsius. And that may or may not be surprising to you. If you find that surprising, The reason being is because all of this work, the original, electrophysiology simulation, was done by Hodgkin and Huxley on a squid. So the default values in neuron are generally representative of what's done in a squid. So my caution to you fundamentally is anybody who is doing anything with neuron, just remember that the default is squid. If you're trying to study a mammal, the diameter is going to be a lot smaller. The axial resistivity is going to be much higher. And the temperature, hopefully in a mammal, is much higher as well. So it's always good. It's always good to be explicit. This is one of the Zen of Python things, right? Explicit is better than implicit. Always specify these things. What is the temperature? What is the axial resistivity? What is the diameter? And then there's no ambiguity. All right, so within a section, what happens? Well, I mean, even if you think in terms of Hodgkin and Huxley, they had an axon of a squid giant axon. They had a squid giant axon in Hodgkin and Huxley, and in that squid giant axon, they had sodium channels, they had potassium channels, and we can insert ionic mechanisms into sections of a cell, and we do it in sections of a cell instead of in whole cells because, and there's lots and lots of research by this, including by the late great Gordon Shepherd was very interested in this sort of thing, the spatial localization of ion channels, right? The cell is not a uniform thing. If you're talking about pyramidal, CA1 pyramidal cells, the density of A currents increases as you move further away from the soma and the apical dendrites. So we allow you to specify all of this stuff on the level of each individual section, and the actual mathematical properties can be specified at even finer resolution. But so if I want to insert, so neuron comes with a bunch of mechanisms. So H.HH is the Hodgkin-Huxley mechanism. That's everything that Hodgkin-Huxley included in their original model. So that's a sodium gating, there's a sodium channel, a potassium channel, and a leak channel. Again, are any of these numbers that are involved with Hodgkin-Huxley appropriate for mammals? No, they were not studying mammals. They were studying squid. But nonetheless, it is common to start out, at the very least, as a toy model with Hodgkin-Huxley dynamics. So if I have Hodgkin-Huxley dynamics, H.HH, remember everything in neuron is H dot. Right, a section was H dot section. Here we have our next example, Hodgkin-Huxley dynamics are H.HH. And I want to put them in a certain section. I can say H.HH insert into the apical, where that's presumably a section. Or if I have a list of sections, I can insert them into that list, or I can insert them into all the sections that I've ever defined. There's another one that I'll show you later that's actually super convenient. If you grab a section, like let's say you have the soma of a cell, then you can do, if soma is the name of the section, then you can do soma.wholetree, and that'll give you everything that's attached to the soma, however far out. So knowing one section gives you a list of all the sections, and then you could just do H.HH.insert into soma.wholetree if you wanted. We can also insert mechanisms one at a time into a single section via apical.insert H.HH. It all depends on whatever is convenient for you, but you can express it in either way. Loop over mechanisms and insert them in a specific section, or have lists of sections and insert mechanisms in them. Doesn't really matter. But the point is, different dynamics can be distributed spatially across, and these are called, we call these distributed mechanisms or density mechanisms. There's no specific point. All of the units, all the where there's the individual sodium channel in these models, all of the units are expressed in terms of per surface area of the cell. Oh, I have a little bit of dendrite came off. All right, so I can specify properties. I can read and write properties of a given mechanism. So if I have on the apical dendrite, wherever is 0.2 far out. So this gives me a segment. Remember, I can select a position on the apical dendrite. This looks like I'm specifying a specific location at exactly 0.2, but for the purposes of numerical computation, when we're calculating on density on these density or distributed mechanisms, this is actually referring to the entire numerical compartment, the entire segment that contains the 0.2 location. But in any case, the segment that contains the 0.2 locations, Hodgkin-Huxley parameters, namely the GK bar parameter, I can grab that, throw that in GK bar, or just as easily I could set GK bar to be a different value if I wanted to. So obviously, in the context of Hodgkin-Huxley, incrementing GK bar is going to increase the potassium conductance, or you could think of this as increasing the potassium channel density. So you want to know what happens with more potassium channel density, increase GK bar. You want to know what happens with less potassium channel density, decrease GK bar. So the converse to, oh no, just kidding. And so by the way, so you can loop over if you want to set something. So this is for one individual segment. Obviously, we rarely want to set things individually. But if I want to loop over all the segments, I can do for seg and apical, and then just set all of my segs. Or I could loop over, again, what we've seen, h.allsec or section.wholetree. You can also sometimes create list comprehensions that are convenient. That's really sort of neither here nor there. Why is this here? Interesting. Okay. So converse to this is a thing known as in neuron as the point process or point mechanism. So these are things like current clamps and synapses are located at specific points. And we'll see examples of those in just a little bit. But in order to actually have concepts to actually be able to run things, the other main thing that you need to know before you can start thinking about running anything and looking at examples, Neuron provides this class known as a vector class. So h.vector. And vectors are like lists. They're like NumPy arrays. In fact, they're interchangeable with NumPy arrays. But in the form of a vector, they're aware of neuron properties. They can monitor, let's say, so this would allow me to record the membrane potential at a specific location on the axon. I can record the time. I could also conversely record the spike times by monitoring a specific location and defining a spike as any time that location membrane potential crossed a given threshold. And we'll see these in the examples, so I don't want to belabor this. But so these are the main types of recordings that we want to do. And fundamentally, then there's the question in neuron of how do we actually make anything go? And there's three main options that I'm aware of for making a cell do something, right? If you model a neuron, by default, a neuron that's just sitting in a dish and not getting any input is not going to be very interesting. In order to run any experiments, whether it's in this extracted neuron or in silico, you need to actually give it some sort of input. So one way of giving it input, you could control the membrane potential by either momentarily raising it to a certain value or running a voltage clamp experiment might be more realistic. You can give it a current clamp, right? So inject current. And instead of doing a voltage clamp, just inject current at a certain amount at a certain location. Or you could also, a neuron could also go by synaptic input. In a wet lab experiment, you could think of this as a puff of glutamate gets administered somewhere. That might trigger some synaptic responses. In neuron, what we do is we create what's known as a net stem, which is a driving object that's a fake, basically, synaptic input. And we connect that to a actual synapse. And why do I keep doing that? We connect that to an actual synapse. And the dynamics of the synapses can be of arbitrary complexity in Neuron. We have examples on Model DB where we have, various forms of spike time dependent plasticity, LTP, LTD, all any sort of phenomenons that you're expected to interested in. How is a GABAergic synapse different from an NMDA? All of that stuff. There's examples of these sorts of models within Model DB. You can go and look them up. Neuron itself comes with several built-in synapse models, but the most generic of them is an exponentially decaying synapse model. It gets input, it jumps up in current, and then it just decays. The amount of current that it generates decays over time. So there's some reversal potential that's associated with one of those, and some time constant. And then all you do is you connect your extracellular simulation to the synapse with a netcon, and then that's sort of the third way of getting a cell in silico cell to go, right? You either set the membrane potential, inject some current, or give it some synaptic input. And with that, I think those are sort of the main concepts that I feel that we need before we look at some of these examples. So let me move on to examples. All right. So our first example here in Neuron is how do I generate and integrate and fire cell? So here I've got an example linking to an eBrains collab. It looks like this. Here's our eBrains collab. And let's just look at it. So in eBrains, I don't have to do anything. It, of course, has Neuron installed. So I open a collab. I can import Neuron, load my unit definitions. Always good to have units around. Makes my code more readable. And I'm always going to want to load the standard run library because it's going to make simulating things easier. All right, so within this, Neuron comes with a variety. I want to do integrate and fire cells. Neuron comes with a variety of integrate and fire models. There's the nFire 1, there's the nFire 2, and there's the nFire 4. I thought I was going to say 3, didn't you? But it has to do with the number of time constants. And so the nFire 1 is a model of an integrate and fire cell that has two main properties. It has a time constant wherein things decay over time. So you give it a signal, it's a leaky integrate and fire. So the amount of current that you have, the amount of membrane potential, if you will, that you have decays exponentially over time with a certain time constant. And it also has a hard-coded refractory period. The other really cool thing about this, by the way, is these integrate and fire cell models that are done with Neuron compute the spike times and their response properties as the synaptic events come in. If there's no new once the synaptic event comes in, the integrate and fire cell knows if it's going to fire or not. By the way, the link to this, thanks Pramod, is available in the chat. So the integrate and fire cell knows at any given synaptic event whether that's going to cause it to fire or not. So it doesn't have to compute continuous time. It doesn't take any compute power to exist if it's not getting any input. The only time an integrate and fire cell has to do any computation at all is whenever it's getting an input. And otherwise, it's just sitting there. And so this allows you to scale up to large models of integrate and fire cells with relatively minimal compute overhead. So the ability to do these things analytically. All right, so there's a time constant and there's a absolute refractory period wherein the cell will not spike if it gets input within a certain period of time, let's say 10 milliseconds, doesn't really matter. But so this is how we define integrate and fire cell. Now, the simplest type anyways. I want to make that integrate and fire cell do something. I'm going to need to give it a stimulus. Now, it doesn't really make sense in the context of an integrate and fire cell to inject current or to give it a membrane potential value. So what we instead do is we have to go the synaptic route. We're going to have to construct a stimuli train. And so with our stimuli train, we can construct this with a net stem. We want a noisy stimuli train. Noise here just means that it's generated according to a Poisson process, which there are certain reasons to think that if you know nothing, that's sort of the best knowing nothing approximation for how cells get input. It's a Poisson process. They happen at a certain average interval. So on average, the events happen every 3 milliseconds, starting at the beginning of time. And I don't really care. I just want it to run for a while, to just keep generating events for forever and ever. So this is how I define a stimulus. That's a random sequence of events. And then I need to connect that stimulus to my cell. Well, I use a netcon object for that. We've already seen that for recording times. But I can use a netcon to connect a network, right? So I want to connect my stimuli to my cell. Okay. Two properties that are important when I do this. What is the weight of the input that my cell is getting? So here I'm setting this to be 0.5. In the context of these integrating fire cells, they're said to fire whenever their total value crosses 1. So a single synaptic input with this takes us about halfway to the cell firing. Unfortunately, 2 is still not going to do it, because at the moment that there's any delay at all, that 0.5 will have decayed a little bit, and then so you'll be just shy of 1. But if I get enough synaptic input in a row, my cell will fire. The other thing that's very important to consider is what is the delay of the network, right? In a real cell, there's an event occurs, a cell generates an action potential. The postsynaptic cells don't find out immediately, right? There's time for that action potential to propagate down an axon. That axon potential then causes the release of neurotransmitter. Those neurotransmitters have to propagate across the synaptic cleft. And by specifying a delay, we can approximate all of that in one number. How long does all of that take? Since it's an artificial input, I'll start out with a delay of 0. But if I was building a network, I would use a non-zero delay. And that's it. That basically defines my model. I have a model, I have a stimuli, and I connect the stimuli to the model. I can set up some recording, and I'll go ahead and create vectors. Remember, vectors are a data storage object. I'll create neuron vectors for recording when the stimuli happens and what the outputs are. And I'll use netcons to do the recording of all of these events. And now it's time to go. So in neuron, there's 2 things that you need to know. There's the finalize function. That's for initializing things. And there's the continue run. That's for running things. And initialize takes a parameter such your initial membrane potential. We don't actually have any cells with membrane potential here. So this, in fact, the number that I put here has no effect whatsoever. But I wanted to put it here because anytime that you are using sections and you do have cells with membrane potential, you are going to want to specify what the initial membrane potential is. And by the way that the mod files are generally written, this is not just setting the voltage, but this is setting all of the other gate variables as if I had been voltage clamped at -65 since time negative infinity. So it puts all the gating variables at steady state. All right, so I've initialized my simulation. I've ran it for 100 milliseconds, and then I can pop up a raster plot. And I'm just doing this with matplotlib. There's nothing magic here, but I can plot. I can make a raster plot up here. Pop up a raster plot was what I was trying to say. And so there we go. So I can look at here. I've got a whole bunch of stimuli, but if I have enough in a short enough period of time, the cell fires. If I get stimuli that happen really soon, it doesn't fire because it's the refractory period, and so forth. And we can generalize. So this is sort of the most basic model that you can do, at least in the form of integrated fire cells and a network. I could, however, generalize this and build a network of integrated fire cells. there's sort of this classic example, and this, I mean, it's very boring in some sense, and that doesn't do any computation. But I could build what's known as a ring network, where cell 0 sends a message to cell 1, cell 1 sends a message to cell 2, and so forth and so on until I'm out, in our case, at cell 9, and cell 9 then wraps back around and sends a message to cell 0. So in Neuron, I have a class for constructing these integrated fire cells. So I'll just make a bunch of them. So here I can make 10 of them, and I'll make 10 vectors for recording all of my spike times. And then I'll just go ahead and configure that each cell gets its properties, a time constant, a refractory period. There's a netcon for recording each of the spiking events. And that's the basic setup of the model, except, importantly, the connectivity. So if I want to connect a network, I need to connect a presynaptic object to a postsynaptic object. They're all integrating fire cells. There's really nothing to it. So I just loop over all of my cells. If you're not familiar with numpy.roll, what it does is it just shifts everything by 1. So that you start at, in our case, this is going to move the last guy to the beginning. So cell 9 rather will send a message to cell 0. So this is going to pair cell 9 with cell 0, cell 0 with cell 1, cell 1 with cell 2, cell 2 with cell 3, and so forth. And then I just build a netcon. I connect the pre-object to the post object. I specify a delay because now I do want to have connections. All right, a synaptic wait. This is overkill, but this guarantees us that the input is sufficient to cause a spike. And then the only other thing that I have to do is I have to remember to store my netcon in a list so that I have all of my netcon objects. And if you're wondering why, this is because of how Python works. Anything that's not anything that doesn't have a way of accessing it doesn't exist in Python, right? It's what's known as garbage collected. So we have to store it so that that netcon continues to have the effects. So we can take that, and we've defined our network. But now again, it's a network. Think of it as a slice sitting in a dish somewhere. It's not necessarily going to do very much unless we give it a stimulus. We can give it a stimulus in the same way as before. It's not very interesting. We can take that, initialize a model, run the simulation, and now when we make our raster plot, cell 0 got the one and only stimulus. It sent a signal to cell 1. Cell 1 sent a signal to cell 2, causing it to fire. 2 sent 3 to fire, and so forth and so on. And I get this cyclic firing pattern. It's a very simple model, but it's the only behavior that we could get out of it. Seeing no questions in chat, I'm just going to keep going. All right.
I think, Robert, if there are any questions, people can just speak out. All right. I think they can unmute and they can just ask here as well.
All right. Seeing no questions in chat and hearing nothing. I'm gonna keep going. All right. So, but Neuron's not really known for its integrated fire capabilities, which I think is too bad because they do play really nice with multi-scale models. So you could imagine that I have some part of my network that I want to study and great biological complexity, and some part that am perfectly happy abstracting away and integrating fires. All of this stuff is, again, Neuron is this unified framework for neural simulation. So you can do that and just build these multi-scale models that focus on complexity where you care about. So if I wanted to, the next thing that I wanted to mention is, so those are sort of integrating fires. That's kind of all there is to integrating fire until we go into types of integrating fire cells. If I want to start thinking about the classic Hodgkin-Huxley that got a lot of computational neuroscience going from the beginning. We have that built into Neuron with the H.HH mechanism, as we've already seen. So again, these are all just running off of the eBrains collab that Pramod linked in the chat. All of these things are going to look fairly similar, so I just want to point out what's different here. If I am trying to do a Hodgkin-Huxley simulation, I'm going to want my units. I'm going to want to load Neuron. I'm going to want to load my run control library. Here, I'm using plotly.graphobjects as Go. Plotly, if you don't know it, it's a graphics library. Nice thing about Plotly is it's interactive, even in these, even in eBrains when you're running it remotely, as opposed to Matplotlib, which is only interactive if you're running it locally. Or at least I don't know how to do it otherwise. Plotly is great for shape plots if you want to look at morphologies in a Jupyter notebook, either locally or on Google Colab. I personally have not had great luck with eBrains on this one. It uses a lot of power, I think. But it works great on Colab, Google Colab. So in any case, so Plotly is just a graphics library. Just going to add that here. But so now if I want to construct a Hodgkin-Huxley simulation, I'm going to define a cell, right? So these are, I'm now talking about something where ion channels are being expressed. It needs to be made of sections, or at least one section. So let's just say that I'm going to have the soma, or I guess if I was doing Hodgkin-Huxley originally, it was the axon. And into my soma, I can insert my Hodgkin-Huxley dynamics, H.HH, and I can specify that my soma has a certain diameter and a certain length. Let's say, so I'm assuming that's 10 microns long. And so there we go. So I've defined my geometry. My cell has one section. I've defined my and I've said how long it is, what the diameter is, and I've defined my biophysics. I'm using Hodgkin-Huxley mechanism. Now that I've done that, I can insert, this is our first example of what Neuron is unfortunately called a point process, or a point mechanism. So here I'm going to have a current clamp. It's a point mechanism because it's inserted into a specific point. So this is a current clamp that's going to inject current into the center of the soma. As I do that, it's going to have a certain amplitude. So this is 0.3 nanoamps. I'm going to delay. So that's, in other words, when do I start injecting current? So I'm going to start injecting current at time 1 milliseconds, and it'll have a certain duration. So I'll inject current for 0.1 milliseconds. And by the way, we'll see later, or if we don't, depending on time, there's examples that are linked in the collab of injecting non-constant currents. So don't think that you're limited to square pulses or anything like that. But that's just the easiest to set up. Setups, define two vectors to record certain locations. So h.reft is recording the concept of time. I can record the membrane potential at the center of the soma. Initialize, now the membrane potential very much does matter. And I can run it for a certain period of time. And then once I've done, I can plot it. So I see that at time 1, I was at negative 65 millivolts. I pretty quickly jump up towards, by 1.1, well, I can't seem to get to 1.1 exactly. Oh yeah, but I can't. But by 1.15, I'm at negative 56 millivolts, and I can just see there's a delay before the sodium channels really start opening, that regenerative activity is happening, and I get the action potential. All right. And so this is our basic generic Hodgkin-Huxley model. I can vary this by adding some random, that's not it. Like I did with the integrated fire cells, I could give it random input. So here, in order to have random input, instead of having a current clamp, I'm going to really want to think about synapses and synapses that are receiving input. So everything is the same. I construct my cell the same, but now I'm going to define a synapse. And the simplest type of synapse model is this exponentially decaying synapse. Again, like a current clamp, that a synapse happens at a specific location. Where does it happen? In our case, it's going to happen at the center of the soma. And that synapse is going to have a reversal potential and a time constant. The time constant, some synapses keep turning on for a long time. Some synapses keep turning on for a short time. Reversal potential mainly has to do with the effect of whether or not it's an excitatory synapse or inhibitory synapse. The reversal potential is like 0 or something like that. That's going to cause the cell, for the most part, to want to raise its membrane potential when the synapse is active. If it was -80, that would cause the cell to want to lower its membrane potential. But so in any case, so now I've defined a synapse. So there's some location on my cell that's going to receive synaptic input. My cell already has Hodgkin-Huxley dynamics. I can construct a net stem, same as before, same as with the integrated fire cells. And again, I really do want to emphasize this. It's, Neuron is this multi-scale framework. We can reuse these components at these different levels of spatial complexity. And then I can use a netcon to connect my net stem, my artificial stimuli, with my noisy Poisson process to my synapse, give it a certain weight and no delay because it's just artificial stimuli anyways, record everything, run it. And then now, for example, once I've ran it, I can see that, so this is the output. Sometimes I get synaptic input that causes a bump, but it doesn't quite bring me to threshold because of whatever reason. It's not been enough in a certain period of time, whatever. So I get a regular firing activity out of my model. But fundamentally, the net stem part and the netcon part is exactly analogous to what we did with the integrated fire cells to get that behavior. Let me shut down the kernels on some of these guys so I don't blow up my memory usage. The other thing that is often desirous, people want to look at parameter sweeps, right? So if I have my model, and again, sticking with the simple Hodgkin-Huxley model, right? So all of this is the same. For some reason, I wrote it the other way. So Hodgkin-Huxley.insert in the soma doesn't really matter. But now let's say that I want to do a current clamp experiment, and I want to know what would happen if I injected different currents. So I can set up my model, right? I set up all of my parts exactly as before. But now if there's something that I want to sweep over, fine. I just sweep over it, right? So for IC.amp and all of these different possible currents, let's just go ahead and initialize and run the simulation for each of those cases. And then at each point, I have a new time vector and a new voltage vector, and I can just plot that. And so I can see as I mouse over this, hey, look, if the amplitude was 0.1 nanoamps, I don't get any response at all, or just barely. 0.2, I see it start to elevate, but it doesn't fire an action potential. And by 0.3 nanoamps, I do get a response, but it takes a while. As I increment the current, the response happens sooner and sooner, but it does seem to be approaching some sort of a limit. And all we had to do, we built our model, and the only thing that we had to do was just loop over whatever it was that we wanted to store. Everything else was exactly the same. One other thing that I want to point out, as we're looking at these sorts of things, Hodgkin-Huxley did not consider ion accumulation. They have sodium currents and they have potassium currents, but they were working in this giant, massive axon, right? So in the neuron version, we're using 500 micron diameters, and you wouldn't expect to see any meaningful change in sodium concentration or potassium concentration. But in smaller mammalian cells, you might. And if you care about that kind of thing, or you care about what's happening on the intracellular level, Neuron provides this RxD submodule for reaction diffusion specification. So here, I define my core of my model, Hodgkin-Huxley dynamics, exactly as before, but now I have to add 3 lines. And so with these 3 lines, I define a region that I care about. So everything that's on the whole neuron that's attached to soma, there's nothing attached to soma, so this is kind of a cheat. But if I had a morphologically detailed cell, this would be important. I give my region a name. This is the cytosol. And I map this to sort of the classical electrophysiological region. That's the I region. We can talk about this if anybody wants, but it's the region just inside of the plasma membrane, right? Because the region, in terms of electrophysiology, all that matters is what's just inside or just outside. But if I'm talking about chemical state, I might care about the cytosol, I might care about the mitochondria, I might care about the ER, and I really do want to be able to specify an arbitrary level of complexity. And rather than try to build in, oh, well, here's an ER case in the neuron and a mitochondria case in the neuron, we just allow you to specify your own regions with their own properties. And there's other options that we're not showing here. But fundamentally, all we have to do to allow accumulation is we just say, well, okay, I'll define sodium to be a species in the chemical species sense, living on the cytosol. It has a certain name, a certain charge, and that's it. I mean, neuron already knew about sodium because it had Hodgkin-Huxley channels in there, and Hodgkin-Huxley channels told it about sodium. But by defining this as part of the reaction diffusion module, what I'm saying is, hey, and I care about these concentrations. They're going to change. I want you to respond to them as appropriate. And everything else is exactly the same. I can set up my synapses and my net stems and my netcons, and I can record things. By the way, if I wanted to record sodium concentration, this is how I record sodium concentration. And what I see is that as my cell is firing, as time goes on, the spikes don't get as high. And you wonder why that is. And that's because the intracellular sodium concentration is rising. Why is the intracellular sodium concentration rising? Because Hodgkin-Huxley, the key part about it is every action potential lets in sodium. And Hodgkin-Huxley doesn't have a mechanism. They didn't define how the sodium was pumped out. They didn't have to worry about homeostasis. But if you're using these small mammalian cells, you do. Only if you care about these things. Maybe you don't care about sodium concentrations and you're perfectly happy having them not be dynamic, in which case, great. Just don't add these RxD terms. But if you do want the dynamic stuff, all you have to do is add these 3 lines and everything is responsive. I have one last example that I want to cover here in my examples, which is neuron is known for morphologically detailed cells. That is neuron's claim to fame. So how do we do a morphologically detailed cell in neuron? Fundamentally, we need to get a morphologically detailed cell from somewhere. And so what I'm doing is I'm going to pull one from neuromorpho.org. Neuromorpho, if you don't know it, is this repository of morphologies. So I'm just going to go ahead and pull a morphology detailed cell from neuromorpho.org. It's C91662.swc. Doesn't really matter. I need one extra library that I need to load, which I load library neurons import 3D library. And so here's an example of how I might use it. I declare a reader, I load in the import 3D, and I pass my reader to my import 3D tool. Annoyingly, it has this underbar GUI name, but if you pass false, it doesn't generate a graphical element. It's the import 3D tool, and then I can instantiate that morphology either at the top level, which is everything that we've done so far has been at the top level, But you could imagine that I want to group things together into classes in Python so that I can have defined, this is my pyramidal cell class. And so then I can instantiate it into a specific object, right? So that I have one pyramidal cell with one morphology and one set of properties. And then once I've done it in an object-oriented way, I can build many, many of these pyramidal cells. So the other thing that I'll point out here, when you do that, it creates a bunch of lists inside of the pyramidal cell. There's a self.all, there's a self.apic, there's a self.den, there's a self.axon, there's a self.soma. So you can access it by regions of the cell, and you can explore all of these things, and they print out really nicely because Neuron does a lot of effort in trying to make sure introspection is possible. But so in any case, let's just we can do that. We can define our cell class, and then if I look at do a dir pyr, this thing that I've created, there's all of the Neuron, all of the like generic Python stuff, but fundamentally there's an all, an apic, an axon, a den, and a soma. And if I want to know what sections are there, I can just look at, well, what is pyr.all? Well, they're .soma of 0, .axon of 0, .dend of 0, blah, 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 .apic. So that's the apical dendrites. And I could grab any individual section if I want by, give me the fourth apical section. Well, there you go. It's pyr.apic of 4. And so using this, I figure out where I want, but then I just do everything exactly as before. Again, we went from point cells to morphologically detailed cells, same exact thing, right? We can construct a current clamp, put it at a specific location, initialize, run, and then just pop up a plot shape. So plot shape is one of two, I'd say, neuron-specific graphs. So here I'm plotting a false color map of voltage at a specific time, specifically at time 2 milliseconds in our simulation. The other main neuron-specific graph that I don't have an example here for you is what we call a range var plot. So imagine I want to know how concentration changes moving from the soma to the tip of the apical. So by defining a path along those 2 points, you can define a path along those 2 points and just sort of plot out concentration as a function of distance from the soma, let's say. And that's often helpful to look at. If you prefer, you can also, so here I'm having my plot shape display using matplotlib. You can also have it display using plotly, and then you'll get an interactive graph, at least if you're running it locally. With that, that was the core of my examples. I do want to touch on a few things. I saw somebody just put their video on. Is there a question or? Okay. Just a couple of brief notes before we go into the core neuron discussion. So we've already seen one of the great sources for us for data is neuron morphologies. There's 235,000 of them now on neuromorpho.org. They're all available in a standardized format known as SWC, which is to say that there's 235,000 that Neuron can just read in without having to do anything. You do want to check. There are certain regions, and you can read the slide at your leisure, but there you may want to be sure to check, do various tests on the cell to make sure that the cell is appropriate for simulation. Not everything is studied with the purpose of simulation in mind, and so they may have made approximations. Only way to know for sure is to read the paper and do some experiments on it yourself to make sure nothing's strange. As you do all of this stuff, you'll generate data. And then so the question is, how do you get data in or out of Neuron? Pandas is a great tool for getting data in and out of Neuron. It's a great tool for any sort of data analysis in Python in general. So if I want to, I have a vector of t times and a vector of voltages. I can just use .2 CSV and dump it to a file, right? So data.csv. And then that's going to give me this comma separated values file. Looks kind of ugly, but if you double click it, it'll probably open in Excel for you, right? So you can take this, you can load this in Excel or MATLAB or whatever it is that you like to do your data analysis in. So you're not stuck in Python. And likewise, you can read values in. You can use values that you read to drive a current clamp or basically any property in Neuron. The other thing that I want to, and there's a collab linked here, but I'm not going to go into it in detail. We can modify. We could also another thing that I find extremely useful if I'm doing longer running simulations, or if I'm running simulations, a series of simulations on an HPC. What I'll do is I'll use SQLite. Comes with Python. It's a database. It's built in. Don't have to do anything, so that's great. And what I'll do is I will have all of my values. I'll do my parameter sweeps. But before I do my parameter, so I'll have my for loop. And then the first thing I'll do inside of my for loop is I'll check to see, hey, did I already do this? Is this already in the database? And if so, then I do nothing. And if it is new, then if it's not something that's in the database, I just run it, and then I have pandas throw the results in the table when it's done. And why would I do this? I do this for two reasons. Well, I mean, the main reason that I do this is because sometimes simulations get shut down. If you're running something on an HPC, you certainly have a wall limit that you've probably had to set, but you could also just imagine, hey, sometimes there are errors in other people, not you, but other people have errors in their code and they don't work. Lots of weird things can happen. So it's nice to be able to have what you've got before anything glitches out. The other nice thing about this, the reason I really love this, is because if I have everything stored in a database, I could just access that database. I could separate the code that generates the data from the code. It gives a clean separation from the code that generates the data and the code that displays the data. And so the cool thing is, if I have a long running set of parameter sweeps and I want to know how things are going, I don't have to wait. I can just have another script ready to go, set up, pull the database, whatever do I have, plot that, and then I can run exactly the same thing a little bit later when I have more data. And I can see where things are going. And this will give me a sense, because you don't want to run it's terrible if you run a long running thing only to find out that your data did not do what you wanted it to do. So being able to making this split, I find, is really convenient for letting me explore the results as they're coming in. And it's easy because Python comes with database, SQL database. So you don't have to even do anything to set it up. It's always there if you have Python. This is just another example of, hey, look, so I have data that's come from somewhere. It's pretty boring, but this is a ramp current clamp, and I can use this to drive a current clamp, or I could use this to drive whatever. And the way that we do this, instead of a vector play, instead of a vector record, we do a vector play. We pass in whatever we want to change and the corresponding time values. And then we basically always pass in true. And the reason for always passing in true is to emphasize that we want to interpolate. But it really, don't worry too much about that. Just always pass in true. And so then this is the response of a Hodgkin-Huxley cell to a ramp current injection. It's firing repeatedly, except the response is, it's just getting so much current, it's never really able to recover. And so the cell ends up in a maintained depolarized state. And so those are sort of the last of my examples. I do want to give very, very briefly a quick shout out to a number of third-party tools. One of the great things about Neuron, having been around for a while, is it does work well with a lot of other tools that exist. So there are libraries that sit on top of Neuron, things like NetPine, allow you to write models for networks in a way that handles some of the abstractions for Neuron. Not really Neuron specific, but I gotta say Get. Get is great. Get is great for remembering why thing. Get is great for avoiding the problem wherein it worked yesterday, but it doesn't work today. Well, let's go back and see what's different. Get. But in any case, there's a number of tools that work with Neuron. So just as a shout out, LFPi, if you're interested in calculating local field potentials, is extremely helpful. The bluebrain project created BluePi Ops, which allows qualitative model fitting. So how is it that, well, it allows qualitative model fitting. How do we decide? We want our cells to fire at a certain rate and have a certain spike width or whatever. BluePi is great for answering those kinds of questions using genetic algorithms to do so. With Neuron, you want to actually measure these things. EFEL is great for this, right? So, hey, I've ran a simulation. I want to calculate the what were the action potential amplitudes, or when did they happen? What were the interspike intervals? What were the spike half widths? EFEL does all of that. Just feed it in your vectors, and it'll calculate this and so much more measures of a spike train, the time series. So just want to give them a shout out, and also want to give a shout out to Elephant, which I think is also presenting at some point during this eBrains conference. Elephant plays a similar role in my head to EFEL for calculating the electrophysiology properties. One thing that I find that Elephant does that EFEL doesn't is it allows you to do some spike train analysis. So if you're interested in things like the Van Rossum distance between two spike trains, or the Victor-Pepura distance between two spike trains. So I have a list of vectors of spike times, and I want to know how far apart they are in the sense of edit distance. So Elephant is great for that. Elephant has nothing to do, by the way. Elephant's just a generic electrophysiology tool. But since we simulate electrophysiology, that means that all of our data can work with their analysis, and it's important to, it's a great thing to be able to exist and be able to take advantage of. And with that, that's my run through